Hello guys, so today we shall be discussing another most important topic regarding a group of fungal infections, right? So all these groups, let us say there are around 4 or 5 groups of fungal infections, all of them are grouped under a single title called as mycosis, right? So they are called as systemic mycosis. So today we shall be dealing with systemic mycosis, Now, regarding this systemic mycosis, few important things all of you have to know. What are the group of fungal infections which are grouped under the systemic mycosis? So, let me mention this very quickly so that we can dive into the topic. So, the first systemic mycosis which I want to tell you is histoplasmosis. One is histoplasmosis. So, this topic is very important for your MBBS exam. So, please pay attention. So, one is your histoplasmosis, right? And what is the other one? The other one is blastomycosis. Other one is blastomycosis. We shall be discussing the pathophysiology or the pathogenesis of each and everything in very much detail. Okay, don't worry. So, the next one is Coccidioidomycosis. Coccidioidomycosis, right? And finally we have to discuss the Paracoccidioidomycosis. The last one will be Paracoccidioidomycosis. So, what are the list which I have mentioned here, guys? What are the list which I have mentioned here? The list which I have mentioned here is these all 4 different types of fungal infections are grouped under the systemic mycosis. So, what are they? One is called as histoplasmosis, another one is called as blastomycosis, the third one is called as coccidioidomycosis, and the last one is called as paracoccidioidomycosis, right? So, we shall be discussing the properties of all of them. We shall be discussing how each one is different from other. I mean, how are they causing the infections, right? Are they releasing any toxins or are they releasing any spores? So, everything we shall be discussing right now. So, please rest back and listen to the lecture, right? Now, all these systemic mycosis, all of them, they exist in 2 forms. I mean, there is an exception, but in generally talking, that these systemic mycosis, they exist in 2 forms. So, if this yeast, the fungi, right, if this yeast is existing in 2 forms, you call it as a dimorphic yeast, right. So, you call this as a dimorphic yeast or a dimorphic fungi. Now, when I tell dimorphic yeast, you just told me that dimorphic yeast means that yeast which exists in 2 different forms. So, can you tell me what are those 2 different forms? Yes. So, what are the 2 different forms are? For example, this yeast lies outside your body. Some of these yeast, they stay in 2 forms, I told you, right? So, what is the first form is they lie outside the body and they also lie inside the body. So, they live outside the body, they live inside the body. When I tell they live outside the body, outside in the sense where? In the soil. So, one form lives outside the body and the other form lives inside the body. One form lives outside the body and the other form lives inside the body. Now, when I tell outside the body, one thing which has to come to your mind is that I am talking about the soil. Can you tell me what is the normal temperature of the soil? It is around 25 to 30 degrees. So, what I am trying to tell you is that at 25 to 30 degrees centigrade, right, at 25 degrees to 30 degrees centigrade, this yeast stays in the soil in a specific form. Okay, and that form is called as mold form, right? So, what is that form called as, guys? That form is called as mold form. What is mold form? I'll explain you in a minute. But before that, I'm telling you it stays inside your body. Inside your body in the sense, inside your tissues, obviously. Inside your body in the sense, inside your body tissues. So, what is the normal temperature of your body tissues? Somewhere around between 36 to 40 degrees centigrade, right? So, somewhere around between 36 to 40 degrees centigrade. So, in the tissues, in what form does this fungi live? So in the tissues it leaves in the form of yeast form, right. So overall, if someone asks you, can you tell me what are the two different forms, right, what are the two different forms in which this fungi can live? Yes, this fungi is called as a dimorphic fungi because outside the body it has one form, inside the body it has another form. Why? Because of the temperature difference. Why? What is the temperature difference? Outside the body in the soil it is somewhere around between 25 to 30 degrees centigrade. So there it will live in the form, it will make its body in the form of a mold, and the same time inside the body in your tissues the temperature is higher enough, so obviously it will be in an yeast form. What I exactly mean to tell is that, let me put it in the form of picture. For example, mold form in the sense, okay, all of these they are dimorphic, right, they exist in two forms. So let me take one example, for example histoplasmosis. So how does this histoplasmosis live? So how does this histoplasmosis lives is that, for example, you see this, what I am drawing here now? Yes guys, what am I drawing? So what I am drawing is, I am drawing a branched hyphae. You know fungi has hyphae, right? So these are called as branched hyphae. So these branched hyphae are nothing but called as the mold form. And where do these mold forms stay? All of you know, yes, they stay in the soil. But the moment if you get infected with this fungi, so obviously the moment it enters into your body, it transforms itself into an yeast form. And how does an yeast look? Yeast looks like this. Yeast looks in this way. What are these things called? These are called as budding forms, right? What forms are these? These are called as budding forms. I can also call it as budding yeast, right? I can call them as budding yeast or budding forms. So if you see a septate hyphae, a branched septate hyphae, what are septa? See these lines which I am drawing, these red color lines which I am drawing right now, these are called as septae. And there are branches. So this is a branched septate form which is also called as a mold form which lives in the soil, and these are the budding yeast which lives in your body tissues. But what is an exception? You told me all these four different types of fungi, right, all of them they live in the same way, dimorphic, except one. Out of all these four, except one, and that is your Coccidioidomycosis. So how does this Coccidioidomycosis live? This Coccidioidomycosis, let me write it down guys. Coccidioidomycosis, it has only one form. Let me draw that form. You see, this is the form. This is the form through which it lives. Now this form is called as spherule. This form is called as spherule form. So whatever I have drawn this is a single spherule. So when you closely, you know, microscopically when you look into that spherule, you will find small spores in that. You will find small spores in this spherule, and you know what are these spores? These are called as endospores. And what are these spores called as guys? These are called as endospores. These are called as endospores. So obviously when you are affected with this type of fungi, these spherules they enter into your body, mainly the lungs, okay. So they break down. These spherules they break down in the lungs and the endospores are released, and those endospores they cause associated diseases, right. We shall be discussing what type of diseases they cause and whatever it is, we shall be discussing. Okay, fine. So you told me that dimorphic yeast exists in two forms, one is mold form and yeast form. So if I wanted to see, should I go to the soil and should I open the body tissues and look at this form? Not needed. Can you grow this so that I can see clearly in the lab? Yes, I can grow this actually. So how do I grow this guys? How do I grow this? For example, how do I grow this mold form? I grow this mold form and this yeast form in a specific agar. Right? For example, the mold form, I can grow this or I can cultivate or culture this in a specific agar, and that is called as saborad agar. Saborad agar. And what about the yeast form? Yes, even yeast form also I can cultivate and grow in blood agar. In blood agar. So these are, you know, some of the important things which every medical student, every MBBS student have to remember for your exams, right? So this is the simple thing which I have told you, right? So this thing is enough regarding the properties of this, the introduction part of this yeast or the fungi. So if we go deep into this, what are the things which we can see? The next thing which you can see here is, having discussed the dimorphic yeast, you should know, and by the way guys, remember this, always, you know, everywhere people love exceptions. Routine things, you know, I know, every one of us know, but exceptions only few know. So the people, the examiners, they mainly concentrate on the exceptions. Okay, so Coccidioidomycosis, they do not stay in two forms, only one form, that is spherule, which has got endospores within it. Now, another important thing is how they are going to infect you. How are they entering into your body? How will they affect you? Right? What is the way of transmission? So when we discuss about the transmission, always remember it is better we make a picture, right? Regarding the transmission, let me draw a simple picture, right? Obviously, you get these fungal infections. These fungal infections are transmitted to you through the spores. You inhale the spores, the infection starts. So how are you inhaling the spores? What is going to happen? I'm just going to give you a general outline, but later on when I discuss about each and every fungi, I shall be discussing it in clearly, right? So right, all of you know, so this blue color thing which I'm drawing from the nose, this is your nasopharynx, and this thing which I'm drawing right now here, this is your oropharynx. Nasopharynx, oropharynx together they form laryngopharynx. So all the way you come down, you have got your bronchi here, right? These two branches are bronchi, and obviously you have ended up in your beautiful lungs over here, right? So how are you going to get infected with this? And by the way, there is no fun in drawing that you have got some blood vessel over here, right? So just for a mark. So let us say that these are the spores. You know, these are the spores. Let us say these are the spores of Coccidioidomycosis. Okay. Now you inhale these spores. Now the moment you inhale these spores, what is going to happen? What is going to happen is that the moment you inhale these spores they get into your body from the nasopharynx, from the oropharynx, right? So as they are passing from the oropharynx, they can cause ulcers also. I'll be telling you later on. So all these spores, all the way they come down and enter into your lungs. So when they enter into your lungs, right? When they enter into your lungs, what is going to happen is that obviously these spores are not digested by our human enzymes. You know, if any bacteria enters into your body, with the help of lysosomes, the hydrolytic enzymes and all, with the help of all the chemical mediators, you can digest that bacteria, you can break down that bacteria. But these spores you cannot break it down. So when you cannot break it down through your own human enzymes, what will happen is that all the cells they come there and surround the spores. Looking at them, the remaining cells, in the sense the neutrophils, macrophages, okay, listen carefully. All the neutrophils, macrophages, they come there surrounding the spore. Looking them, the remaining macrophages, they come there. Looking them, the neutrophils, they come there. Looking them, the lymphocytes will come there. So all of them, they come there, but they can't do anything. Instead, what will happen is that they stay there for a long time. All these cells, they stay there for a long time, try to release some kind of enzyme, some kind of chemical mediators, and finally you will find an abscess, right? You will find a local infection. So that local infection will stay for a long time. That is called as a chronic condition, chronic pulmonary condition, and that is what is called as a granuloma. Okay, so granulomatous diseases are always chronic. You see granuloma formation in a chronic condition. For example, let us say I work in a cotton factory, right? If I work in a cotton factory, or let us say sugarcane factory, or whatever it is. So I have been inhaling a lot of particles. Okay, let me give you a perfect example that I work in a coal factory. So obviously I inhale coal. So when I inhale those cold dust particles, are they living or non-living? Obviously non-living. So when a non-living material enters into your lungs, looking at that, all your macrophages, neutrophils, right, eosinophils, lymphocytes, all of them, they come there, try to kill that non-living coal particle. But can they kill? Can you kill a non-living material with your human enzymes? No. So in that case also, you will find a granuloma formation. So can I tell like this, that in almost all occupational lung diseases, you find a granuloma? Yes. So anyways, I'm just giving you a brief output of all the things which are happening. So it starts from the mouth. In the mouth it can cause ulcers, it will enter all the way into the lungs, and in the lungs it is going to create a havoc, and it can cause a granuloma formation. Right? That is why we call this as a chronic lung disease. This is as a chronic lung disease. And second thing that can happen is, guys, what can happen is that these spores, they can even enter into the blood circulation. These spores can even enter into the blood circulation. And from the blood circulation, where do they go? They go all the way to your liver, or sometimes they go all the way, let us say, to your spleen. They go all the way to the liver and spleen and infect the liver and infect the spleen. So you're going to end up having hepatomegaly, you're going to end up having splenomegaly. Not only that, even these spores, they go to the joints and infect the joints, leading to arthritis. So overall, you understood how these spores are entering into your body and how they are causing the infections. So this is the basic thing which I wanted to tell you. Right. Regarding the transmission, the source of each and every fungi, we shall be discussing now. Right. So we shall be discussing, you looked for all these 4 different types of fungi previously, right? Like Histoplasmosis, Coccidioidomycosis, right? All these are not fungi basically. All these are the diseases that are caused by the fungi. What I am trying to tell is, for example, if you look at histoplasmosis, histoplasmosis is a disease, is a fungal infection, and this fungal infection is caused by Histoplasma capsulatum. This is caused by Histoplasma capsulatum. In the same way, you came across Coccidioidomycosis, right? Coccidioidomycosis. So Coccidioidomycosis is mainly caused by Coccidioides immitis. Coccidioidomycosis is mainly caused by Coccidioides immitis, right? And can please remind me, which form does it stay? Dimorphic or in a spherule form? Obviously it is in a spherule form. The third thing is Blastomycosis. Blastomycosis. Can anyone tell which form, what is the fungi that is responsible to cause Blastomycosis? Blastomycosis is caused by Blastomycosis dermatidis, right? So it is basically caused by Blastomyces dermatidis. The next thing is Paracoccidioidomycosis, right? Paracoccidioido, these names are really lengthy. Coccidioidomycosis. So Paracoccidioidomycosis is caused by Paracoccidioides brasiliensis, right? So it is caused by Paracoccidioides brasiliensis. So these are the 4 important things which you have to know. So one is caused by Histoplasma capsulatum, the next one is Coccidioides immitis, and the next one is Blastomyces dermatidis, and the last one is Paracoccidioides brasiliensis. So these things you have to know. Why? Because these can, as usually, these can be an MCQ, right? These can be an MCQ. The next one is the source of this infection. How are you going to get? For example, let us see that this histoplasmosis, whatever is there, right? So histoplasmosis is mainly transmitted through bats or birds excretion, right? So they are mainly transmitted through bats, birds or bats excretion. Birds or bats excretion. So how am I going to get exposed with this? Those who love to go into caves, right? Those who love to explore, those who, the trekkers, mainly the mountain trekkers. So if they are resting in the caves, if they love to, explore the caves and all, so in those cases, caves are completely filled with the bats and all, right? So through those bats excretion, when they smell, the spores can enter into their lungs and cause this histoplasmosis. The second thing is Coccidioidomycosis. And how are you getting transmitted? I mean, what is the source of this? Through the earthquakes. When there is an earthquake, these spherules are blasted out. Okay, so during an earthquake, these spherules, whatever are there, they blast out. When they blast out, the endospores are released and these endospores are inhaled. So basically, the earthquakes. Next, regarding the blastomycosis, so blastomycosis is most commonly seen in the patients who are having HIV, right? And finally, the paracoccidioidomycosis. You see paracoccidioidomycosis in patients who are always contact with the soil, who are always in contact with the soil. So these are some of the important things which can make an MCQ for you. These are the important things which you have to remember. I mean, if they wanted to give you a clinical question, so they will be giving like a patient came to the clinic complaining of so and so infections, right? So when we have taken an X-ray, we came to know that there is some chronic lung infection in him, right? He is also having pain in the liver, I mean the right hypochondric region, pain in the left hypochondric region where there is spleen. And he gave me a very beautiful history that 2 to 3 weeks back he went to explore the caves, right, on a mountain trekking event. So he went to explore the caves and there were many bats in that. He shooted those bats. It was so good, fine, but the thing that has unfortunately happened is that he smelled, right? Obviously you take breathe inside the caves, right? So when you take breathe inside the caves, the excretory material, the spores in that, they get inhaled into your lungs. You inhale them basically and you end up in the hospital having histoplasmosis or the remaining things, right? So this can be a question. I mean, they'll these things like birds, bats, excretion, earthquakes and also they'll be giving it in a history. When you look at the question, in that question, the patient's past history, in that past history, they'll be giving you these things. So they are not giving you as an MCQ, instead they'll be putting them in a form of a history. So you have to know all of that. Right. So let us start regarding the pathogenesis now. We shall be discussing the pathogenesis and each and everything. So let us start with the first bug, the first yeast, that is histoplasmosis. So before discussing the pathogenesis, so let me draw a simple picture, right? So I'll draw a picture and then I'll explain you the pathogenesis of this histoplasmosis, right? So let me draw a simple picture. So he is a patient who came to me, right, complaining that he is having some sort of problems with his respiratory tract, and all of you know that he is gonna have a beautiful nasopharynx. He is also gonna have an oropharynx, right? So he is having his laryngopharynx like this, and all the way down, all of you know that he is gonna have his own lungs. Have his own lungs. Now let me even draw a blood vessel here, right? And all of you know that this blood vessel, let me draw two organs here, right? So let us say this blood vessel is supplying his liver. At the same time, this is also supplying his spleen. Now look here. Regarding these spores, what important thing you need to know is that these spores, this guy is inhaling these spores, and at the same time, let us say the spores, even while inhaling, he opened his mouth and the spores also went through his mouth. So when the spores enter into his mouth, as I already told you, these spores in the mouth can cause some ulcers, right? So these ulcers, they are named as oral mucosal ulcers. These ulcers are called as oral mucosal ulcers. So he is going to have oral mucosal ulcers, and next thing is that as he is inhaling the spores they all the way enter into his respiratory tract and finally enter into the lungs. So right now the spores are in his lungs. Okay, now the moment spores enter into his lungs, what are the things that are going to happen? First thing is, see these points guys, these points which I am drawing here, look here, these points here, these are called as hilum of your lungs. Right, these are called as your hilum of your lungs, where your lymph nodes are located. These lymph nodes are called as hilar lymph nodes. So as the spores are passing there, they even touch these hilar lymph nodes and cause infection in the hilar lymph nodes. So hilar lymph nodes will be swollen, and that is what is called as hilar lymphadenopathy. So patient is also going to have hilar lymphadenopathy. So apart from that, patient is also going to have hilar lymphadenopathy. Third important thing, now the spores are within the lungs. Now the moment spores, when they enter into the lungs, what is going to happen is that, all the different types of, let us say, neutrophils, right? Not only the neutrophils, even your macrophages, all of them, they come there, they release some enzymes, they release some, let us say, inflammatory mediators or whatever it is, and macrophages, they ingest that, they release the hydrolytic enzymes. So overall what is happening is that a kind of fight is going on there between the spores and our immune cells. So the result of this fight is that you find a small abscess that has been formed here. So patient is also going to have an abscess, lung abscess, right? So there will be a cavity that will be formed in that abscess. Abscess is nothing but a cavity, right, containing all the pus. So there will be a cavity that will be formed. So can I tell, in these patients, when they inhale the spores, they're going to end up having cavitatory lung lesions? Yes. So these patients going to have cavitatory lung lesions or cavitary lung lesions. Cavitary lung lesion. Second important thing that can happen is that, as there is infection that is going on within the lungs, all these immune cells like neutrophils, macrophages and all of them, they come there and they sort of create a fight over there and form a lung abscess, I told you, right? Along with that, a lot of fluid also will be released as a result of this inflammation, as a result of this infection or the inflammation which is going on in the lungs. A lot of fluid will be released and that fluid will be deposited in the lungs. So can I call this condition as pneumonia? Yes. So patient, whenever he is having any kind of lung infection, he always will end up having a condition called pneumonia, right? Fifth important thing is that I already told you these spores can even enter into your pulmonary capillaries or the basic blood vascular system. Now the moment spores enter into your blood vascular system, what is going to happen? Now these spores will travel all the way in the blood, it enters into the hepatic artery, and finally the spores get lodged in the hepatic artery, in the liver. Through the hepatic artery they are coming and they enter into the liver. So once they enter into the liver, they cause inflammation of the liver, right? So what do they cause, guys? They cause inflammation of your liver. So your liver will be swollen up, right? At the same time, these spores can even enter into your spleen and cause inflammatory changes in the spleen. So there also will be inflammation that is noted in the spleen. So patient's spleen will be also swollen up as a result of inflammation. So when there is swelling of liver and spleen, you call this condition as hepatosplenomegaly. Hepatosplenomegaly. So all these are the conditions which you see basically, right? So you're gonna see an oral mucosal ulcers, you're gonna see hilar lymphadenopathy, you're gonna see a cavitatory lung lesion as well as pneumonia in the patient. Not only that, you will also see hepatosplenomegaly. Now, out of all this, two things you have to know, that this patient is having pulmonary problems All the problems which I have written, you can group into two. Patient is having pulmonary problems, patient is also having systemic problems, like problems with the systemic circulation and problems with the systemic organs like liver and spleen. So that is the reason why these fungi not only affect your lungs, they also affect your systemic organs. Wherever there is systemic blood supply, they also affect your systemic organs. That is why these fungi are grouped under systemic mycosis. Okay, right. Another important thing, the most important thing I'm going to discuss now, in the lungs, apart from cavitatory lung lesions or pneumonia or hilar lymphadenopathy, a very special thing that is going to form in the lungs, that is granuloma. How a granuloma is formed in a lung with the help of the spores. Look here, the moment the spores enter into the lungs, obviously within your lungs there are macrophages, and these macrophages of the lungs are called as alveolar macrophages. So let me draw one alveolar macrophage here. So let us tell that this thing which I am drawing here, this is your alveolar macrophage, right? So this alveolar macrophage has got its own nucleus. Now let me tell that this is a spore over here. Now these alveolar macrophages will engulf that spore. Look here, these alveolar macrophages are engulfing that spore. Now once they engulf that spore, the spore enters inside, and this process is called as phagocytosis. And the resultant product which is formed here, this is called as phagosome. Now once a phagosome is formed, guys, what is going to happen is that these, now lysosomes will come. I'm not going into much detail which is not needed exactly, but you have to know the pathophysiology. Now the lysosomes enter into this phagosome, right? Slowly the lysosomes come. Okay, let me draw a simple picture. So these green color things are called as the lysosomes. So these lysosomes, they come and fuse with the phagosome and form phagolysosome, right? And these lysosomes, after forming this particular phagolysosome, look here, after forming this particular phagolysosome, what is happening is, from the lysosome, all the hydrolytic enzymes, they enter into this phagolysosome and they break down this spore. Now once they break down this spore, the spore particles whatever are there, this macrophage will pick up from its body inside. Let us say I am a phagolysosome, I am putting my hand inside my body and I am finding where is the spore particle and I am lifting it up and I am showing it up, right? So the same thing that is happening here is, once the spore gets broken down into multiple pieces, one of the spore particle is picked up by this alveolar macrophages, and this alveolar macrophage will show up this particle with its own hand, and that hand you call it as MHC class 2 molecule, that is major histocompatibility class 2 molecule. So you got a spore here, a spore particle. Now why this macrophage is showing and to whom it is showing this thing? It is showing to T helper cell, right? So now your T helper cell comes all the way here, it examines here. What is going on with this? What is this particle that my friend MHC class 2 molecule is showing? And suddenly this T helper cell recognizes that this is a foreign particle, and immediately, what does it do? This T helper cell starts releasing, so it is releasing two important cytokines. One is interleukin 2 and interferon alpha. You know what is going to happen? This interferon alpha will go all the way like this, it will go all the way and activate your macrophage. Now once this interferon alpha activates your macrophage, and this macrophage is going to convert into a special activated macrophage, and that is what is called as epithelioid cell. Okay, so basically how does an epithelioid cell look is that it looks like a squamous epithelium, right? All of you know columnar epithelium are like pillars, squamous epithelium are squeezed epithelium, right? I'm not going to draw that squeezed epithelium because that thing does not support with my pictures over here, but with the explanation. So I'm just going to draw the simple macrophage but little big. Okay, so this is the macrophage over here and you have got your nucleus. Now this macrophage right now which I have drawn, these are not cilia, okay? Don't think this is cilia. This is a representation of an activated macrophage. This is a representation of activated macrophage, and this activated macrophage, my dear friends, This is called as epithelioid cell. Epithelioid cell. Right. So what is a good thing about this epithelioid cell? The simple thing is that this epithelioid cell is big. Point number one. Point number two is it has got a lot of cytoplasm. When you compare a normal macrophage with this epithelioid cell, normal macrophage has got little amount of cytoplasm. As this activated macrophage or epithelioid cell is big, so it can accumulate lots of cytoplasm. And the nucleus is also pretty big, okay, when you compare with the normal macrophage. So this is an epithelioid cell. Now what is going to happen is that, so this is one single epithelioid cell. So in that way, many epithelioid cells, they join together. Okay, so this is a single epithelioid cell. Let me draw a few more epithelioid cells like this. I'm not going into much detail here. So not detailed picture as well. So there are multiple epithelioid cells which I'm drawing now, right? So multiple epithelioid cells have got its own nucleus, and multiple epithelioid cells have got its own cytoplasm. So all these epithelioid cells, they join together. They join together, and how does the resultant picture look, guys? Look here. All these epithelioid cells, they join together, and this is the resultant picture. Look now. All these epithelioid cells join together in this way, and after joining, each epithelioid cell is having one nuclei. So when all of them join together, there are many nuclei here, right? So this is called as a multinucleated giant cell. This is called a multinucleated giant cell, okay? So this is called as a multinucleated giant cell. So from macrophage we formed epithelioid cell. Epithelioid cells fuse together and form multinucleated giant cell. And look what is happening. Within this multinucleated giant cell, all these nuclei also fuse together. So when all these nuclei fuse together, how is the picture going to look? Let us say this is an epithelioid cell, I mean the multinucleated giant cell, and all the nuclei, they fuse together and form a very big nucleus. This very big nucleus is a macronucleus. And what is the shape of this nucleus? The shape of this nucleus is like a horseshoe, right? So the shape of this nuclei is like a horseshoe. So can I call this particular nucleus as a horseshoe nuclei? Yes, horseshoe nuclei. But what is the name of this cell? This cell is called as Langerhans cell. And what is the name of this cell, guys? This cell is called as Langerhans cell. So what is happening here? What is happening is the first thing which you can see, which you can appreciate here is you're gonna have Langerhans cells in the center like this. There are Langerhans cells in the center, right? Surrounding that Langerhans cells, you're gonna have epithelioid cells like this. There are epithelioid cells in this way. And one thing we forgot, you know what is that? You forgot to mention the spore in the center. So there is a spore. Surrounding the spore you have Langerhans cells. Surrounding them you have got epithelioid cells, and surrounding them you have got your macrophages. I'm just writing it as M. M stands for macrophage. So surrounding them you have got your macrophages like this, and surrounding the macrophages, what do you have, guys? Any idea of a granuloma formation? Very important for your MBBS. Surrounding them you have got your lymphocytes. What lymphocytes? B lymphocytes. You have got your B lymphocytes like this. So there are multiple B lymphocytes surrounding that. So look how beautifully the layer has been formed, guys. In the center you have got your spore. Surrounding that you have got your macrophage, the Langerhans cell, and surrounding the Langerhans cell you're gonna have your epithelioid cell. Surrounding the epithelioid cell you have got your macrophages, and surrounding all of them you have got your B lymphocytes. So this entire unit which I have just drawn in front of you, right, this entire unit is nothing but called as your granuloma. This is called as your granuloma. Such granulomas are formed in the lungs. I am writing it as G. G stands for granuloma. You see, such granulomas are formed on either sides in the lungs. So whenever you see a granuloma in the lung, can I call this lung condition as a chronic lung disease? Yes. So another important point I have to add here is I can also see a granuloma. So overall guys, if I wanted to make a bigger picture and tell you overall, what are the things which you appreciated here? One is granuloma formation, right? The next one is you got hepatosplenomegaly, right? The third one which you have got is pneumonia in the patient. Not only pneumonia, you have also got hilar lymphadenopathy. LAN stands for lymphadenopathy. Hilar lymphadenopathy, and what all things still I forgot. What all things which we saw? And yeah, oral mucosal ulcers, right? You have also got oral mucosal ulcers. So all these conditions, all these conditions are very beautifully associated with your infection called histoplasmosis. And histoplasmosis is caused by Histoplasma capsulatum, right? So this is the basic thing, and histoplasmosis is caused by Histoplasma capsulatum. So fine, so this information regarding the pathogenesis is enough, but if a patient is coming to me, when I examine him, I find all of these things, and I have a doubt in my mind that the patient might have histoplasmosis, so I will do a lab diagnosis. So what is a lab diagnosis which I do? Obviously, I will take an X-ray. In the X-ray, I can find bilateral hilar lymphadenopathy. I can also find pneumonia there. I can also find a lesion, a dark lesion that is called as a granuloma over there, right? And still what I can do? I can do a histopathological investigation. So when I take a histology, when I take a slide of this, you know what will I find? What will I find is that this histoplasma, the budding yeast of histoplasma, the spores or whatever it is, they are located in the macrophages. So that is what I can find, right? I'll show you the histopathology now. I hope all of you can see this slide. You can see this entire unit is your macrophage. Within that macrophage, you can see small particles here, round particles. So this is nothing but called as your histoplasma. So macrophage engulfing or histoplasma within the macrophage is the characteristic appearance which you can see in case of histoplasmosis, right? So what is this specimen about? This specimen is about histoplasma in the macrophage. Histoplasma in macrophage. So this is the thing which you can see. Okay, so this is a very important thing. Histoplasma engulfed by a macrophage is the pathognomic sign or is a histological diagnostic importance of this thing. Okay, right. So we are done with histoplasmosis. Now let us enter into Coccidioidomycosis. Okay, so let us enter into Coccidioidomycosis. Nothing more guys, nothing special thing about this. The same things which you have to know, the same things. So let me draw a picture again and you have to tell me now what are the things that are going to happen. So this is the skull. Right, and you have got your nasopharynx. Now you are going to tell me what is going to happen with this patient who is affected with coccidioidomycosis, where spherules are released, right? Within those spherules, endospores are there. So patient is going to have his own lungs over here, right? And let us draw a blood vessel over here. So this is the basic blood vessel. Now tell me what is going to happen. The patient will inhale the spores. I mean, I am discussing right now the pathogenesis of coccidioidomycosis. So patient is going to inhale the spores, and these spores all the way they travel down like this, and finally they enter into the lungs. Now once they enter into the lungs, what things you can see guys? Obviously you can see, yes, tell me, you can see pneumonia, you can see pneumonia, you can see a lung abscess, you can see a lung abscess, you can see a pneumonia, you can see a chronic lung lesion, which is also called as a granuloma, right? And apart from this, you know what all things you can see? I mean, all the pulmonary complications, all the pulmonary diseases which are associated with this fungi are same. Even you go for histoplasmosis, coccidioidomycosis, paracoccidioides, and all of them, the lung lesions are same. But what is unique? I wanted to discuss the things which are unique. The unique things are, once these spores, by chance they enter into your circulation. Now look here what is going to happen. Let us say these spores are traveling all the way in your circulation. And let me draw it a little bit more. Let us say this is your blood vessel over here. And this blood vessel is supplying to two different parts in this way, right? Let us say this artery over here, the artery which is on the top, this is supplying, let us say, your skin, okay? So this artery is supplying your skin like this. Now what happens when it supplies to your skin, I will discuss right now. And next thing is that, let me draw another picture of your, what is this picture, guys? Tell me, what am I trying to mention here? I am trying to mention what? A joint, right? Any joint it can be. So look here, once these spores, they come all the way in through the circulation, and from the circulation, the spores, if they manage to enter into the skin, you know what are they going to do? They are going to initiate the same chemical reactions. They are going to cause inflammation there, they are going to cause a havoc, and finally what is going to happen is that in the skin over there, you're gonna find a lesion which has a bumpy elevation like this. This is called as erythema nodosum, and the surrounding area is also reddish because it makes the entire area reddish like this, right? So this condition, guys, is called as erythema nodosum. Erythema nodosum. And another skin disorder which these spores are going to cause is that erythema multiforme. This is going to cause erythema multiforme. So two important things to take into consideration. One is erythema nodosum and the next one is erythema multiforme. Okay. Next thing, if these spores descend down and they enter into the joint, okay, if they enter into the joint, what is it gonna cause? Obviously it will gonna cause arthritis. This is gonna cause arthritis to the patient. Okay. Apart from pulmonary infections, what are the systemic infections? Arthritis, erythema nodosum, erythema multiforme. Not only that, for example, if this infection, guys, let me draw another artery that is supplying to your brain, okay? If this infection goes all the way, I mean, if these spores travel all the way like this into your brain. So once they enter into your brain, and what is it going to cause, guys? It is going to cause meningitis, right? So overall, what are the things that it is going to cause? The first thing that is going to cause is meningitis. What are the things which we discussed in Coccidioidomycosis? Meningitis and the pulmonary complications, right? I'm not writing all of them. I've written in histoplasmosis. The same things apply here. Pulmonary infections. Apart from pulmonary infections, this is also going to cause erythema nodosum. And it is also going to cause erythema multiforme and arthritis. So these are the things which you basically encounter in a patient with what? With coccidioidomycosis, right? Fine. So you told me that if I wanted to diagnose histoplasma, I will look at a histological slide and I see macrophage engulfing all those histoplasma. So that is a characteristic for histoplasmosis. What I am going to find for coccidioidomycosis is I am going to find spherules, right? The blasted spherules. For example, if this is a spherule over here, right? A blasted spherule, from this spherule I am going to see the spores that are coming out like this. So these are the spores that are coming, and what are these spores, guys? These are called endospores. So this particular picture which I have drawn here, right? This particular picture which I have drawn here, this is a diagnostic importance. This is the histological slide which I will find basically, right? The next important thing we have to discuss is what? The next important thing which we have to discuss is blastomycosis, and we should also discuss paracoccidioidomycosis. Paracoccidioidomycosis. So in case of blastomycosis and paracoccidioidomycosis, what is going to happen? Are there going to be any kind of pulmonary infections? Obviously, there will be pneumonia basically. Apart from pneumonia, is there any unique thing which you can tell us? Yes. Even the spores, they affect the skin on your face, right? So the spores, they affect your skin on the face, they affect the facial skin, leading to a specific type of lesions on the face, on the moustache here, in the mandible region here on the face. It is going to you know, create some lesions, and these lesions are hard and they are bulbar in shape, round hard lesions. These are called as verrucous skin lesions. These lesions are called as verrucous skin lesions, right? So what are these lesions called? These lesions are called as verrucous skin lesions. So how does a verrucous skin lesion look basically? So this is an appearance of a verrucous skin lesion. As all of you can see here, how does a verrucous skin lesion looks, right? You see on the moustache region all these things which you can see here, right? Let me highlight this. All these things which you can see here on this is nothing but a verrucous skin lesion of your blastomycosis. As well as paracoccidioidomycosis. Right? So this is how you diagnose. And second thing is that, okay, you also told me that in paracoccidioidomycosis you see verrucous lesions. Yes, okay, I see verrucous lesions, but how am I gonna classify? I mean, how am I going to differentiate it? Because in blastomycosis and in paracoccidioidomycosis, both of them I find verrucous skin lesions. So which one is para and which one is blasto? How am I going to know? So how we are going to know is that with the help of a histological slide. So when you take a histology of this para part, I mean the paracoccidioidomycosis, so this is how you're going to find. So this picture here which I'm drawing, which I've mentioned here, this is the paracoccidioidomycosis, right? Why it is called para? Because you see the budding yeast surrounding the ring over here. So this looks like a ship wheel. I mean, this looks like a captain wheel ring, right? Captain's wheel. Captain's wheel budding yeast, right? So in the same way, the picture is not given, but in blastomycosis, what type of histology will you find? That is broad based budding yeast. What is that name? That name is broad based budding yeast. So overall guys, I'm not going to write it down, but you have to remember it, that in histoplasmosis, what is the histological finding which you will find? You will find, I mean, in microbiological slides, they will give you a slide, they will put up a slide in front of you and ask you, what is this? So for histoplasmosis, you will find the histoplasma engulfed in the macrophage. For coccidioidomycosis, what do you find? Yes, for coccidioidomycosis, you find spherule releasing endospores. And for blastomycosis, you will find broad-based budding yeast. And for paracoccidioidomycosis, you will find a captain wheels appearance. So this is very easy, I think. Paracoccidioidomycosis diagnosis is pretty simple. Thank you so much. So this is the end of the lecture.